When you look at single neurons in the cortex, which is the outside sheet of the brain of an animal, you see that those single neurons are not quiet all the time, but they have like a single burst of activity, and they can either happen all the neurons together and then there's going to be a period of silence, or like it can happen that there's no real pattern between them and like there's independent activity bursts in the single neurons. When you look at what actually happens in the brain of an animal, you see that the activity lives in like anywhere in that spectrum. So there can be periods of like very synchronized spontaneous activity, that's how we then call it, or like desynchronous spontaneous activity, where all the neurons independently have bursts of activity.
There was a lot of previous work showing if you present sounds when the cortex is synchronized, how well are they represented as compared to situations where the cortex is desynchronized. But that hadn't been done in the context of an animal actually making judgments about the representations of those sounds.
We tried to address that functional link by asking mice to make a perceptual discrimination. So we played one of two simple sounds to them. One was a high frequency tone, the other one was a low frequency tone. And depending which sound we played, the animal had to report at a right or at a left response spout. And we had the animal perform this task while we were recording the activity of the auditory cortex.
The desynchronized state is traditionally seen as a state where the animal orients its cognition towards the environment rather than internally. So our hypothesis coming into the study was that during those desynchronized state, when the cells are playing their own tunes, the next decision the brain makes is more likely to be accurate.
Contrary to our expectation, we found that the answer was no. That in general, knowing whether the cortex was synchronized or desynchronized didn't help us to predict better whether the mouse would make a mistake. And so we were somewhat puzzled by this finding, but then Davide had the insight that maybe we should look at how the mice had been doing before, whether they had made a mistake in the previous trial or whether they had been doing well for a while.
So I repeated the analysis and separated the trials in which the animal had made a mistake in the previous trial from trials in which the animal had made a correct choice in the previous trial. And we found that in trials in which mice had made a mistake in the previous trial, our intuition was correct. The animal would make more accurate decision if the cortex was desynchronized before the sound was presented.
Now, our current hypothesis is that there are two ways in which mice can do well. One way is through actually focusing attention on the environment. And we think that errors trigger that strategy. But in the conditions of our study, where the stimuli are not very difficult for the mice to categorize, they can also do well in a sort of automatic fashion.
It seems that when the animal is doing the task well and in a flow state, kind of like being in the zone, like a musician lost in a performance, completely absorbed in the melody, the auditory cortex is not heavily involved. But when an error is made, the brain recruits the auditory cortex to help perform the task. So now the state of the auditory cortex influences the animal's behavior.
And so we're doing experiments now where we try to selectively interfere with the activity in the auditory cortex, and we want to know whether the effect of those perturbations is higher after errors. If our hypothesis is correct, that's what we should see.